I appreciate Dr. Bellin for coming today. Thank you very much. And we'll get started now with our webinar and welcome our esteemed guests. Today we have a very wonderful mix of lab research translating into clinical research and therapeutics for long COVID. And I am very honored to welcome our two wonderful guests. Dr. Schamess has um, been a guest at our webinar before. And Dr. Schamess is an internal medicine physician at The Ohio State University. He's one of the lead physicians at the long COVID clinic at Ohio State University and is doing cutting edge work on long COVID therapeutics. He is also a co-host for the long COVID clinical podcast. Welcome, Dr. Schamess.
Thank you,
And,
Anita.
and then we have, it's my honor to welcome Dr. Amal Amer. Dr. Amer is professor microbial infection and immunity at the Ohio State University. She is vice chair for translational research, microbial infection and immunity. Her research actually is fascinating. It focuses on understanding the interplay between the inflammasome and autophagy pathways and host pathogen interactions in the lung and the brain. So, for someone like me who is not a lab researcher, what it means is that she is developing pathways for newer medications and understanding conditions like long COVID. So, she holds the torch for newer drugs that will help us treat these patients. Welcome, both of you.
Thank you, Anita, for this great introduction. I'm so excited to be with you guys today.
So, Amal, we'll start with you. Your research explores the intersection of, the autophagy and inflammasome signaling, as we talked about. How, just to set the stage for you to talk about your research, how might dysregulation in these pathways contribute to the lingering immune dysfunction we see in long COVID patients? How do these pathobiological mechanisms contribute to what we see as long COVID symptoms?
So thank you. This is a great question. And it's not just my lab, several labs in the US and around the world are trying to understand the underlying mechanism of long COVID. And as we all know now, long COVID affects so many organs. And we think that the pathology is interrelated between the different organs. However, our lab, because we worked before on inflammation in the lung and inflammation in the brain in the context of Alzheimer's disease, our focus here is on long COVID in the context of the brain. And we have a very specific interest in the brain because the number of people who suffered from brain fog was really staggering. And personally, it is a personal interest to me because unfortunately, I got COVID 3 times. Although I stayed home, I didn't go anywhere, but every time I step outside my house, I get it. So after the second time, I got what we call now long COVID. So when I got the COVID, it wasn't severe. Yes, I was in bed, but I didn't go to the ER or anything. And then I felt I'm recovering. I'm physically okay. And then I decided to go back to work. At the time, we were all during the pandemic working by Zoom. And I opened the Zoom. I'm listening to my students. They are explaining their data and their findings. And I realized I can't follow. Once they are finished with one idea or one result and they go to the next, I ask them, oh, why didn't you do this experiment? And they go, Dr. Amal, we just told you we did. So, and it keeps repeating. So I start getting worried and anxious. And then also during the time I realized when I try to read a paper, I can't just follow what is written. I read a couple of sentences and I keep reading them again and again and again. My brain cannot comprehend or connect the ideas. And And the weird thing about the brain fog is that if you're around the house doing your normal thing, you will not feel you have anything wrong. So it's not like Alzheimer's, it's not like dementia. It's that inability to do the very deep thinking, to interrelate the ideas together. And from our work on the inflammasome, we have several knockout mice, and knowing several molecules and how they work, we kind of realize, okay, we have this molecule because it does a lot of important functions. At that time, we knew it's doing these functions during bacterial infection. The hypothesis was is going to do the same functions in SARS-CoV-2, and the knockout of this mice will be protected. And this is how the whole work on the inflammasome started.
Thank you so much. And I'm very excited now, and I will ask you this question later on in the webinar about your research. But I want to shift gears and talk about the clinical side. And Andrew, I'm so excited that I want people to hear about the wonderful work that you are doing. There is an exciting project you are starting on amantadine, and you are trying to understand its effect in long COVID and to see if this medication works. Can you tell us about amantadine? Why amantadine? And what are you proposing to do? And what kind of results do you hope to see?
Yeah, sure. Thank you for the question, Anita. I should just mention, by the way, because I don't know if you said, Amal and I are both at Ohio State University, and we know each other, and it's kind of an interesting story how our lines of work connected. So maybe we can touch on that a little later. But the amantadine story starts long before I met Amal. I'm sure parts of this will be very familiar to other people who are in long COVID clinics trying to take care of patients. When we started in 2021, we had nothing, right? I mean, there was no evidence, there was no data, there was no science on how to treat these patients. And they come into your office and they say, I have these horrible symptoms. I can't think straight. I can't do my work. I can't remember new things. I'm struggling. I'm exhausted at the end of the day. I mean, and all the other symptoms as well, like dyspnea and dizziness and chest pain. But I think it was the brain fog that really kind of caught my attention as being like something, just how disabling and how disheartening it was to patients not to have their brains working. Um, I had done some work before that with traumatic brain injury patients, and it struck me as the symptoms that these long COVID patients were describing were very similar to my brain injury patients. Word finding difficulty, executive function, difficulties, losing their train of thought, sometimes kind of personality changes, going from being extroverted to being very shy because they're embarrassed about their disability. Um, and in the brain injury world, amantadine is often used to treat this, along with some other medications we can touch on later. So I just borrowed amantadine kind of empirically. If it works on brain injury patients, maybe it'll work on patients with long COVID with the same symptoms. And it's a very striking experience, as some of you may know, when you have a patient who's really afflicted with a symptom and they say, wow, that medicine you prescribed me was a game changer. And this is very much like confirmation bias. There's a risk of confirmation bias here because you think of the patients who really did well on this, and you kind of filter out the ones who didn't do so well. But I went on prescribing it, and at a certain point, I kind of accumulated just by chart review our data, and about 50, a little more than 50% of patients had had really good outcomes. So I thought maybe this really could be something. And around that time, there was there was a donor. I heard by word of mouth that there was a donor who wanted to make, fund something in long COVID, and that the higher ups in the university were kind of taking this donor around. And so I started banging on doors saying, hey, we have a long COVID clinic here. Why don't you talk to the clinicians? We have some worthwhile ideas. And they ignored me and ignored me and ignored me for a long time. And then finally, it turned out the donors, they couldn't interest them in basic science. They couldn't interest them in this and that. What they really wanted was to fund a treatment trial. So finally, I got to talk to them and I said, why don't we try amantadine? And they bit, and so they donated some money so that we could actually do a controlled clinical trial of amantadine. And so it's just for people in the audience who are kind of trying to get scientific research started at their institutions. It's kind of interesting how these things actually happen. A few more just kind of interesting things about the study. In addition to the fact that it worked in clinic, there was an article published in Nature, which pointed to a possible mechanism for amantadine, which is amantadine is an NMDA inhibitor. And so why should inhibiting NMDA receptors have this impact on patients? Possibly because we know that tryptophan metabolism is out of whack in patients with long COVID. We know that it's because of this inflammatory state that the brain and the body are in, it tends to be routed down this kynurenine pathway, which then produces, when glial cells, when the brain is inflamed, it produces a compound called quinolinic acid, which is neurotoxic, and it's neurotoxic at the NMDA receptor. So is it possible that these NMDA inhibitors are actually neuroprotective because they're blocking that receptor so that there's less damage from quinolinic acid? And there was a paper that suggested that the quinolinic acid levels correlated very closely with cognitive impairment in patients with long COVID. So we then had a potential mechanism to study. And then the last little piece that's kind of interesting is if you look at the data, there's two things that happen cognitively in long COVID. People have cognitive impairment, which many of people don't even recognize. Like possibly we all lose a few IQ points after we get COVID. Not everybody is symptomatic. And then you've got this group that has very symptomatic brain fog, not all of whom can be shown to have cognitive impairment. So is this really one phenomenon or is this two different phenomena? And if amantadine impacts it, which one will it impact? So we set up this trial partly to just to test blinded placebo-controlled pilot trial of 60 patients to test amantadine against placebo. And we built in testing the kynurenine pathway and the quinolinic acid levels. And we built in both objective sort of cognitive testing as well as subjective symptomatology and a bunch of other kind of patient-reported measures. So that's what we're doing. We've got 57 of 60 patients recruited. So hopefully we'll finish this study, by August or September, and then we'll have some results to share. So that's it in a nutshell.
And Andrew,
Andrew,
how much of can you talk about the dose and such?
Oh, yeah, sure. Yeah, the standard dose for amantadine is 100 milligrams twice daily if people decide they want to try this in their clinic. A few cautions. First of all, it's very teratogenic, so don't use it in patients who are at risk of pregnancy or thinking of pregnancy. Number two, you got to be careful in patients who have a history of any kind of impulse control disorders, alcoholism, gambling, addiction, things like that. Number three, we have found out that a small number of patients will bump their creatinine, which actually hadn't been reported, but I've seen it twice now in our trial patients. So be careful in patients who have chronic kidney disease. And then number four, twice a day, but you've got to do, first dose has to be in the morning, second dose has to be in the early afternoon. If you give them an 8 a.m. and 8 p.m. dose, they will 100% have insomnia. Like it's a very stimulating medication. So don't let them take it at night, either like 8 a.m. and like 1 p.m. or maybe just give both doses in the morning. 200 milligrams a day is the standard dose.
I think
Andrew,
Hector has
very,
a
very
question. Yeah.
fascinating.
Thank you, Hector.
It's kind of interesting because I saw this patient and she taken Robitussin and it helped significant with the recovering from the crash. And
That
Robitussin
makes sense.
has similar mechanism in MDA receptors.
Yeah, that's fascinating. That's really
So
interesting.
it's kind of
Proof of
interesting to see
concept.
this kind.
I love that.
Yes.
Thank you, Hector.
And the other things I saw this kind of patient is a device now they are using in putting the ear, try to looking for the pulsations on the arteries, coronary arteries, as well as the blood flow. And she showed a correlation between blood flow and brain fog. And I wonder if the amantine has some impact in blood flow in the brain.
Perhaps.
Yeah, so it's something
That's
like
what
I
I'm not
was kind
testing,
of,
but yeah.
for the study. Yeah, but no, it's great job. I'm kind of very pleased to hear about it, and I'm proud about you, what you done.
Well,
Thank you.
thank you.
That's awesome. So, Amal, now let's talk about your research. Please tell us about what prompted this, what are you looking for, and how exactly are you, uh, what methodology are you using with the mouse models?
So when the pandemic happened, of course, many of us were asked to stay home unless you're working on SARS-CoV-2. And I had a very excited student who decided to go ahead and try testing SARS-CoV-2 on we have a very good number of inflammasome knockout mice. So the one that I picked was the caspase 11 knockout. And caspase 11 is a important member in the inflammasome complex, and it hasn't been well studied like caspase 1, but we studied caspase 11 in the context of bacterial infection. And what we learned about caspase 11 made it the ideal target for SARS-CoV-2 because caspase 11, of course, mediates the release on the inflammasome. It mediates the movement of immune cells towards the inflamed or infected organ. And also we found that caspase 11 plays a role in coagulation. So you need caspase 11 for the platelets and the blood clots and the endothelial cells, all of them, and even for the complement to come together and make the blood clot. And that is fascinating regarding the question that Dr. Bonilla just asked about the blood supply to the brain. Blood clot in SARS-CoV-2 is a very important side effect, and we found it's also mediated by caspase 11. So we went to our colleague, a virologist, and we learned how to do the infection with SARS-CoV-2. And the virologist said, well, everybody came to me with their knockouts, and your knockout is going to be, is going to get more sick, and that's it. And I told him, no, my knockout is going to be protected. And it was. When the mouse doesn't have caspase 11, the mouse was protected from the severe infection and the acute infection and the cytokine storm during the acute phase. And then after that, we started to look at the coagulation. So we realized that the caspase 11 knockout mice do not have coagulation. And we did that coagulation assay in a very cool way, where we had a fluorescent gel we injected in the blood vessels, and it's fluid as long as at 37. Then you sack the mouse, you put it on ice. Now this gel becomes solidified. You sack the mouse, you take the organs, the brain or the lung, and you clarify them. And now you have the whole blood circulation shining with a fluorescent color. And whenever there is a blood clot, there is an obliteration and there is a dark spot. And you have a computer program that can tell you how in the wild type you have so many dark spots because they have so many thrombi along the circulation while the caspase 11 was protected. So after that, we realized, okay, we have a great model, and caspase 11 is an important candidate drug target. But still, we wanted to look at brain fog. And several other people published stuff related to brain problem. So they infected the mouse, they waited a few weeks or few months, they did some analysis to some pathways, yes, they're there. But how can you tell that a mouse has brain fog? We were just describing, you lose the ability to think and to follow cues. And all the behavior tests that are available for us that we've been using for centuries for the Alzheimer's disease are not at that level. Everybody knows the Y maze and the water maze and all these mazes. They are not at that level. They just measure mainly the memory. If the mouse can remember something he learned the day before. However, we got access to an amazing computerized behavior equipment called the IntelliCage, which is from the Intelligent Cage. And luckily, it's the same donor that helped Andrew with the clinical trial. he was very interested in finding a drug and a treatment for long COVID, and he donated for that expensive machine. With that machine, you put the mice and you put a chip on the back of each mouse, and then the whole cage is computerized. And then you have a program that will make the mouse learn how to get the water. And the mouse will need to do several steps in order to get to the water. But now we're going to change it the next day and do in a different corner and different directions, etc. So what is this machine is testing is exactly what I was suffering from, the cued memory, the steps, that one step will lead to the other and the next and the next. And this is what we were missing in the field to diagnose a brain fog symptom. And we did the test, we infected the mice, we waited 3 weeks, 3 months, 6 months. And even after 6 months, the mouse that got infected cannot figure out and go step by step to memorize how to get to the water. And this is, of course, in comparison to a mouse that never got infected. And we were able to find that indeed they have the brain fog. Why is that so important? It's very important because now, like Dr. Schamess was saying, we need a drug that works in long COVID. And there is a huge line of drugs in the pipeline. And to do a clinical trial, it takes a lot of money, a lot of time. How do you prioritize? How do you know where to start? And we think that the mouse model we have is the best screening method we have right now, because you can use these different drugs to these mice, and the drug that will give the most promising response, where the mouse does not lose the ability to think and do sophisticated memory or sophisticated steps, that's the drug you can start with in a clinical trial. And the idea of caspase 11 is that the mouse that didn't have caspase 11 was protected from all that, was protected from the cytokine storm. The lung were protected. When you see the lung tissue in the wild type and the caspase 11 knockout mouse is nice and night and day. The wild type is all inflamed, obliterated, exudate, and the caspase 11 knockout mouse infected with SARS-CoV-2, the lung is so clear as if it never got infected. So with the brain, when we did RNA-seq, it was just the same thing. The caspase 11 knockout mouse, no changes. However, the wild type mouse, even 6 months post-infection, and I would like to remind you that the mouse clears the virus in 1 week. So imagine 1 week, and then you wait 6 months without any live virus in the mouse, and still the brain show you differences. That's why we think that model with that system is something that the field was really waiting for, and is going to be very useful to screen many drugs in a very short period of time.
That's very fascinating. So, Amal, if I understand it correctly, using this machine, not only are you understanding that there is what comprises brain fog, or these mice have problems with sequential thinking, but you are also able to test out medication. So if there are, trials on medications for newer therapeutics, perhaps something like Andrew is doing, then that can be tested as well. This is really fascinating, and actually it gives us a lot of hope, especially for long COVID, but other infection-associated chronic conditions as well.
Absolutely.
So very fascinating. So Andrew, this brings me to you. I know that you and I attend several meetings together, which are about which also have peers from other parts of the country, other parts of other long COVID clinics. We hear about therapeutics, we hear about repurposed medications, about medications which are off label. What are some of those medications that you find interesting or exciting, and that you want to not only find out more, but you also would like to try for your patients?
Yeah, so I would say, I mean, in some ways, apart from the amantadine, which might be, I might be a little less commonly used. Apart from that, I kind of use the same medicines mostly that everybody else uses. I mean, I use some low dose naltrexone. I use modafinil as if amantadine I think is good for cognitive, stuff. If it's just plain fatigue and you need to treat fatigue, then modafinil is good. I guess I would say, I mean, I often focus first on non-pharmacologic things, and we can talk about that a little later. But, I think sleep is a big lever in patients who have chronic fatigue and brain fog. So there's various medications. I happen to like amantadine as a sleep aid, just because it's kind of long lasting and has some anti-pain effects. I've gotten very interested in the whole dysautonomia aspect of long COVID. So apart from behavioral remedies, there's sort of a branch of stuff you can use if they have low blood pressure, where you're trying to expand volume and raise the blood pressure. There are also those patients with hyperadrenergic POTS, who tend to run high blood pressures, and then you can't use midodrine or fludrocortisone or medicines like that. Um, I've had some success using pyridostigmine, which people may know. It's mestinon, and it's used in patients with myasthenia gravis to just increase acetylcholine at the nerve terminals. But acetylcholine is the preganglionic neurotransmitter for both branches of the autonomic nervous system, and it's the postganglionic neurotransmitter for the parasympathetic branch. So it seems to kind of strengthen autonomic function in general, perhaps a little more in the parasympathetic system than the sympathetic system. But the thing I really want to say about all these medicines, which anybody treating long COVID will know immediately is true, is that a lot of this stuff, when you're trying to treat patients, you are throwing spaghetti at the wall. I mean, you always have to say, look, I'm going to try a medication on you, which I think might work. It works with some patients. It has these side effects. It might or might not work with you. And then you wait, X number of months until they report to you that either it's helping them or they hate it, or it's not doing anything. And then you try something different. And that whole trial and error can be very time consuming, sometimes very aggravating for the patients. If they keep getting medications that don't work or give them side effects, they start to lose hope. So we really need a better approach to trialing these repurposed medications, and ultimately also medications that hopefully will be more targeted to the specific mechanisms of long COVID, which is where Dr. Amer's idea is genius. And I mean, I know she explained it, but I'll just kind of reinforce that, I mean, instead, I mean, a clinical trial, as I am finding out, as I'm leading my first one, is really expensive and really time consuming. And I mean, it'll be 3 years from when we started this to when we publish our data, right? Whereas if you can run this in mice, I mean, you can get this done in a matter of a few months. You can get a sense of whether this is going to work. And obviously you can't just extrapolate from mice to humans, but at least you can get, filter out which medications are more more worth testing in humans or not. And you also, because you can grab hold of their little brains once they die, you get some sense of what it might be doing in the mice, and you get some sense of, okay, are there biomarkers that might help you? And the thing that particularly, I mean, I guess you could, in any mouse model that you can give COVID to, you could treat them and then dissect their brains. But what's really unique in this, like, how do you know if a mouse has brain fog? I mean, you can't ask it. But so Dr. Amer has designed with the IntelliCage this model where you really can, in this very granular way, test their cognitive function and find even subtle cognitive deficits, so that you really do have a mouse model that you can test neuroactive agents on. So I think I mean, she's developing this, and there's still some work to do before this is really ready for prime time. She can talk more about that. But I think this has a potential to really accelerate research in this field.
That is awesome. I think Jackie has a question. Jackie, please go ahead and unmute.
Hi, thank you so much. Yeah, I'm Jackie Becker. I'm a neuropsychologist, and I'm at the Icahn School of Medicine at Mount Sinai in New York. I've met some of you at some, mutual long COVID events. But I was so curious to hear about these studies, and Andrew, your RCT in particular. I'm curious about inclusion criteria, primarily because I was one of the PIs for Recover Neuro, and the way that we designed the study was sort of to be inclusive. And we tested a few different, it was a five-arm multi-site trial. So we tested a few different interventions for cognitive impairment. But I find that very often some of these trials are failing because the inclusion criteria is not particular to having a sort of sub-threshold level of cognitive impairment at baseline, right? And so then there's very little to improve upon during the intervention itself, and so we end up seeing negative results. And so that was my question number one. And then question number two, I'm curious about amantadine because, we've seen it for treating things like depression, and, it can indirectly like lift mood and help with motivation, which, could then also improve perceived brain fog. So when thinking about the mechanisms, like how is that taken into account? And I asked that because, one of our arms also for Recover Neuro was transcranial brain stimulation. We did TDCS and, specifically targeting the, prefrontal areas. And so that was another, problem as well, is sort of like, well, are we helping patients who potentially have depression and brain fog, which goes back to your point about mechanisms and how to properly treat, these patients too, where there may be phenotypes, right? Where we have brain fog due to neuroinflammation and a few different things. And then we have brain fog due to, the secondary and tertiary effects of long COVID. In other words, maybe somebody who has POTS who then has, mood involvement and so on and so forth. So not to say that it is, psychological and etiology at all, but rather just that tends to confound these sort of treatments when we think about mechanisms. So anyway, these are just thoughts that were coming up as we're talking about this.
Yeah, well, thanks for the question. And yeah, I have to say, I mean, you're right. These are knotty problems, and we might very well run into the same difficulties that Recover did. In terms of how we screen them, we use this little, just like 5-question cognitive screening questionnaire that we pulled out of the PROMIS inventory the NIH has. And so if they score anything less than a 5, which basically we're just asking in the broadest sense, does your cognitive functioning affect your day-to-day life, or is there a noticeable decrease in your cognitive function? That's kind of the initial screen that, along with meeting all the just sort of safety, inclusion, and exclusion criteria, allows them to be in the study. We have a whole battery of stuff we're doing, and as I mentioned, right, I think there may be somewhat different mechanisms and different phenotypes for cognitive impairment, which you would measure on cognitive testing. So we're using the NIH toolbox to do cognitive testing versus the whole subjective, sense of of cognitive impairment. And for that, we're using the PAOFI, Patient Assessment of Own Functioning. We're using a more extensive set of questions from the PROMIS patient-reported measures, and we're doing the PHQ-9. But I'm kind of wary of the PHQ-9 because some of the questions on there really will
They overlap.
They overlap, yeah. So
Yeah.
we're also using this depression in what is it called? Depression in the medically ill inventory. And one of the little sub-studies will be how well do those two correlate with each other. And then as far as bioassays, right, I mean, our proposed mechanism is that as the NMDA inhibition is, is responsible for is neuroprotective in patients with high quinolinic acid. So we're measuring quinolinic acid levels at 4 different points during the study and correlating that with both the cognitive function and the response to amantadine. And so we'll see if that hypothesis turns out to be correct. You're right, amantadine does all kinds of other things in the brain, being kind of dopaminergic stimulant and so forth. So it's possible that, if it's helping in some completely different way. So that, I guess, we'll hope to uncover to some degree. So I hope that at least goes some way toward answering your question.
Yeah, no, it does. Absolutely. Thank you. And sorry that I'm off camera. I'm on the East Coast, so it's, almost
It's
9 o'clock here, and I just had to put 3 kids to bed by myself. So
Oh my goodness,
I'm not presentable.
you're a hero.
But I really appreciate that, and I think the studies that you both are doing are incredible, and it's certainly helpful. We have an AHRQ funded long COVID clinic as well, that we're constantly wondering about treatments. Like we recently heard about a few things about famotidine. I guess there was a trial out of the Middle East that had some promising results, and it's a
Yeah.
relatively benign drug. So I don't know if you all have experience with that, but but really quickly back to the inclusion criteria. I think the issue that you may end up running into is because you are including people with only subjective cognitive complaints. Part of the issue that we had with Recover, and the manuscript should be coming out soon, hopefully, is we included people with subjective cognitive complaints, but when we did their objective testing, impairment levels were very low, right? And so, whether the amantadine is going to be helpful for subjective cognitive impairment is most likely going to be what you're testing, right? Because if people are average at baseline and you give them amantadine, right, like average on the NIH toolbox, and you give them amantadine, they won't increase in that, right? And so, does that make sense what I'm saying?
Yeah, I see your point. Right. So right, and we don't. I mean, also there's no premorbid data, so you don't know if they've.
Exactly.
Yeah. So it may be harder to move the needle on the cognitive
Right.
testing, which is okay with me. I mean, if in some sense the subjective symptoms are every bit as much worth treating as the objective.
Totally, absolutely. Yeah. Anyway, just very curious about that research. We had a few pilots that were privately funded as well. So
Yeah.
I hear you on the foundation grants. Those are great.
But I appreciate the advice.
So I have a question regarding this point, that the impairment testing really, you don't see much difference whether before or after the trial. My question is, are these the right tests? Because I had the brain fog, and the simple questionnaire, I don't think I'm gonna do bad on it. It's just, I think we need to think about the different types of tests to
Yeah,
really recognize the
so
brain fog.
you're right in that our tests are designed for, detecting dementias and traumatic brain injuries, and sometimes they don't always capture the more subtle brain fog. But even COVID aside, pre-pandemic, we know that objective cognitive impairment doesn't often align with subjective cognitive complaints, right? Like those things are discordant most of the
Yeah.
time. Even again,
Yeah.
pre-pandemic, 70, I would say, percent of the time they don't align. And so what's tricky is like it's the same thing with measuring, fatigue and insomnia, kind of, right? Where like you have people who have insomnia and are unable to sleep, but even when that improves, they may still feel fatigue, which suggests that fatigue is something separate. It's not sleepiness. It's a different construct. And I think about that when I think about brain fog, is like cognitive impairment is defined as an impairment on tests. And yes, it interferes with functioning. When you think about brain fog, yes, it can interfere with functioning, but we don't find it on our tests. So it's possible our tests are imperfect, and we can adjust for that by simply moving the threshold, right? Instead of using, they're normally distributed, so instead of using like 1.5 standard deviations below the normative mean, we can use 1 standard deviation below the normative mean. But the reality is, like, for a lot of people with brain fog, they're still average. And sometimes it's also particularly because of the fluctuating nature, right? We're capturing people on their good days, and they're able to put forth full effort when they really sit down and try and rest. But the reality is that that's not reflective of their day to day. So it has poor ecological validity as well, right? Because even though they can sit in a clinic and take a test after they've rested for 2 weeks, day to day, they can't exert that much effort, or they experience crashes, or, anyway, so there are a lot of issues that are inherent to cognitive testing. But to me, in these trials, I've seen this constantly, especially with long COVID and with others as well, but trials that are trying to improve cognition without recruiting people with actual objective cognitive impairment. And so it's the same thing with like medication adherence. If you go to a clinic to recruit patients, who are non-adherent, you're by definition missing those non-adherent patients because they're in clinic, right? So like if you then do an intervention to improve medication adherence or, adherence with appointments, you're sort of missing the very critical patients that you should be capturing. So anyway, it's all just food for thought and lessons that we're learning along the way. And so I'm just always curious to hear how people are sort of putting together these studies.
On that note, what I think one way to potentially kind of hedge your bet in that sense is to kind of do more longitudinal and at-home testing to kind of see maybe if you can catch people on their off days. And I know this has come up for other diseases. I think of, trying to catch lupus outbreaks for biomarkers. Like it's different, but kind of the same idea applies. And then it's this very kind of transient, cyclical, but completely random event that you're trying to capture. And so I wonder, or I'm kind of curious what the cadence was for sampling in the Recover study.
Yeah, I mean, we don't do home visits. Those tend to be really costly. We had patients come in for in-person cognitive testing before the intervention, but we did the interventions remotely. So the interventions themselves were meant to be more accessible, and we were in 85 sites across the U.S. So it was, it would be otherwise have been nearly impossible, I think, to implement. But I think you raise a great point, and I think we've talked about that a lot in like the MECFS world too, right? Where like doing at-home testing and capturing them on certain days. But the question then is, do we want to capture them on a good day, right? Where then that's just reflecting their good day and they may just come out as average or normal. Or do we want to capture them during a crash, during a bad day when, they're having an acute brain fog episode? So you know what I mean? Like it's just a hard thing to sort of, I think that in MECFS that like people are talking to about how to measure post-exertional malaise, and do they want to, induce post-exertional malaise and then measure the next few days, or do they want to capture them, when they're not in an episode? I don't know. Like, I don't think that there's a perfect answer, but you're right. I think thinking about all of those things is just really important.
I think, Jackie, you make like two very important points here. Is one of the criteria for include people in the trials. I think we need to be selected. What kind of symptoms we're going to evaluate it, and because otherwise we start including people with a different kind of symptomatology. At the end, the result is going to be negative results.
Right.
And the other thing is like we're doing a study now, almost finished kind of recruiting. In the crash studies, we try to induce crash patients, patients with long COVID, and we're taking samples before, during, and after the crash.
That's awesome.
The cognitive function too, of doing like a cognitive assessment at different times. And so this is going to give you more in clarity about this kind of problem, because you see in the studies, you see the cytokines, different studies show different kind of profiles, and sometimes they are not even close to each other. And the problem is the variability of cytokines in the same patients. This one paper, I think, came from Stanford years back. Jerry Younger, this colleague, collect blood samples from the same patients every day for 25 days. So every day they draw blood, and they look at the cytokine profile from the same patient. It's a lot of variability in cytokines. And they found out a correlation between, they mentioned 51 cytokines per patient. And they found leptin correlate with fatigue, and they found out when the fatigue was worse, the leptin level was high in those patients, like a straight correlation of the cytokine levels and fatigue. So it's something like it's very important when you do a study to select. I think we are wasting too much money because we rush to do the study without sit down and try to select the inclusion criteria and what population we're going to test.
I totally agree. I think you make such great points. And I've been working with Jared Younger. Actually, I'm the chair for the cognitive work group for the NINDS MECFS Common Data Elements Committee. It's a mouthful, but we've been working on these things a lot, and it's fascinating to me to think through all of these details, and also using objective endpoints, I think, is really critical. But there was a study, I think it was last year maybe, on long COVID related to cortisol levels, and they found I think low cortisol or something in long COVID. But if you look at when they tested, right, it was like different times of the day for different people, when we know that there's variability, diurnal variability in cortisol levels. So things like that it's just it's thoughtless. And so I completely agree, and I think there's so much that needs to go into it.
It's kind of interesting about the cortisol levels. I agree. I look at the data, the collection was at different times, and we collecting cortisol levels in people in the morning between 6 a.m. to 10 a.m. and just one single patient have a low cortisol. Majority of the people was normal.
Yeah.
We tried to submit a letter to the editor, but they didn't accept
Oh,
it.
you should have.
They didn't accept it.
I tried to submit one to the New England Journal of Medicine too, where it was the Hampshire paper, about patients losing IQ points, and it was like, oh my God, it was so poorly done too. From the cognitive side, that was a whole other thing. But it's a problem, that people can't reply to these these high impact papers too.
So Andrew, I'm looking forward to read your paper.
Yes, we are all
Yes, I'm
anxiously
telling you,
waiting.
Thank you, Hector.
Amazing.
Thank you.
And
And
Amal's too. Amal's paper too.
Very
Yes.
fascinating
Yes.
stuff you are doing. Well,
Thank you
thank you.
very much for this enlightening discussion. Nick, do you want to wrap up?
Sure. I just want to thank everyone for attending, and I just want to remind everyone that we have videos of our recordings up on the YouTube page for our group, and we look forward to seeing you at the next one, and look out for a video recording link of this session in the coming weeks. So thanks everyone, and we'll see you next time.
Thank you.
Thank you all so much.